Thank you, Rohini, and thank you, everyone. This is my first time, not to Britain, but to this meeting. I feel a little bit guilty talking, since I'm not going to be talking about genomes. But I will be talking about pain. I want to thank all the organizers, and a particular note to Lucy Criddle, who straightened me out on many different ways. So I don't know if she's in the audience, but please send her my best. I'm not jet lagged yet, so I'll be fine. And I like beginning with this first slide. Just the lights are a little bright, but they'll hopefully come down a little later. These are covers of the journal Pain when I was editor-in-chief. The only good thing about it is you got to pick the covers. Because if you reject the paper, then they hate you, and if you accept it, it's because the paper was spectacular. But you do get to pick the covers, but it illustrates the complexity, and I think that's what we're here all about, the complexity of the pain experience. It's not just about the injury. I'll come back to this example. It's a remarkable one where this is someone with CRPS, complex regional pain syndrome. And you can't see pain, but he's describing his pain. He's a sculptor, is having scalpel blades in his foot. And that's what we're trying to understand. Mechanisms, of course, trying to treat. So this is the title of the talk. It's part of what I'm going to talk about, most of what I'm going to talk about, but there's other things I wanted to introduce because we've been trying lots of different things lately. I get bored trying the same thing. And so I'm going to be talking about three different areas. And the first, I'm going to be talking about the brain, looking at work with some postdocs in the lab on pain affect. I'm going to talk about the discovery of novel analgesics with some chemistry colleagues at UCSF. So a totally new direction that we've taken. It's been pretty exciting and productive. And then talk about what the title of the talk is, long-term imaging. And I think it's entering a new field, a new way to process how pain is generated. So looking at pain affect, well, we know that there are at least two different dimensions of the pain experience. We know where the stimulus is. There's a sensory discriminative dimension. It's localized. It has a modality, and it has an intensity. And there's an affective and a motivational component. That's what pain hurts, and that's what makes it special. Otherwise, it's just sensation. And understanding the distinction in how the brain processes. Pain is a percept. It's not a stimulus. What I always tell the medical students. The pain is not in your hand. They say, well, sure, it seems like it. No, the pain's actually a product of your brain. And so where are these percepts generated? Well, imaging studies have, in fact, identified that different areas light up, and we hear about the term, which I happen not to like, the pain matrix. We can come back to that. But fMRI and EEG studies have definitely identified regions. And we know, for example, in a simplistic sense, that the sensory discriminative component, where and the intensity, is located in somatosensory cortex, among a few other places. And the affective component, that emotional experience that makes pain so special and problematic, is in part in the process in the anterior cingulate cortex, as well as in the insular cortex. And we know that based on imaging studies, but also based, in fact, even in patients on ablation studies. If you destroy the anterior cingulate cortex, you can reduce or eliminate pain affect. But these approaches, they don't really explain how the ACC handles pain affect. It's a big blob that you cut out and everything's just fine. But we're interested in trying to understand it. And so we, by accident, to be honest, turned to general anesthetics. And we were starting calcium imaging. I spent my life studying descending control systems, as many of you know, working on spinal cord and working on afferents. And finally, someone, in fact, years ago, my first teacher, Ron Melzak, when I was an undergraduate, said, Allan, you're wasting your time studying spinal cord. That's not where pain is. You're not going to help anybody. You better start working on the brain. So I finally listened to him 30 years later. But we decided to start doing calcium imaging in the brain and see if we can make sense of it. And make a long story short, we were having trouble working with this with general anesthetics, and it turned out the anesthetics turned out to be actually interesting, and we literally moved into studies of how general anesthetics work. And the first one that's interesting is nitrous oxide. Now, you may be familiar with nitrous oxide. It is still used as a general anesthetic. This goes way back. It's a wonderful cover from Anesthesia and Analgesia. The amazing nitrous oxide. What's so amazing about it, of course, when I first left England and came to San Francisco, and he'll kill me for telling you, Howard Field invited us to his house, and one of the rooms had nitrous oxide to introduce people. There were a few other things going on. Remember, this was the 70s. It does make you laugh. But it's used in dental procedures, it's used in labor, and it still exists. And we have been studying its effects in mice, in the setting of pain, to see what happens. And just to give you an example, so here it's a little bit, if we can have the lights down a bit, the animal is breathing either air or nitrous oxide on this side, and they're choosing between warm and hot. And you can see pretty clearly what happens. The warm guy, the air guy, goes over to the hot side and pretty much stays away. The one breathing nitrous oxide, and this was about 60%, he'll go over, come back, go over. We don't know what's going on in his head, but clearly he's not avoiding the hot side the way the air is. So it gives you the impression that there's an emotional piece that's missing. We can also see this using reflexive measures, which is a little more objective perhaps, and we monitor different behaviors in response to, say, a laser. We can do it with formalin or a laser, and we do withdrawals, shakes as index of a reflex, as opposed to a lick. And I remember Chufuma, I hate the term, but a good friend, but he refers to the animal coping. Talk about euphemizing. The animal's coping if it licks. Fair enough. So now we're looking at the animal in a controlled situation, breathing air in this case, and different intensities of a laser are hit to the paw, and you'll see different behaviors. Shake and a lick, there the animal clearly licked, so we would consider that an emotional response. That's just no response, a withdrawal, etc. You can see all the different types of behaviors one can get. But clearly, when the animal licks, we see that as an index that something got to the brain, there was a percept, and that it was unpleasant. And you can score these and see what the effect of nitrous oxide is. And what you see that on reflex measures, there is a significant effect of the nitrous oxide, maybe a 10% drop. But if you actually look at licks, it basically goes to zero. And it's highly significant. So nitrous oxide clearly is predominantly taking out the affective component of the experience. That was interesting. Well, if that's the case, that nitrous is blocking affect, then in a sense it's doing what an ablation of the anterior cingulate should be doing. And one would predict if you now monitored activity of the anterior cingulate and expose the animal to analgesic doses of nitrous oxide, you would expect that it would shut off the anterior cingulate. Well, let's see what happens. So we put grin lenses over the anterior cingulate, a recording in a highly controlled chamber. Temperature is controlled, et cetera, especially important when you're dealing with general anesthetics. We're monitoring, this is usually synapsin-driven, although we use other selective agents to look at inhibitory interneurons. Don't have time to get into that here. And we monitor lots of neurons and calculate them, and let's see what happens. So here we have just monitoring the anterior cingulate. Oh geez, that was supposed to work. Can you turn the lights down, please? There we go. So the nitrous oxide is increasing, and clearly in contrast to expecting inhibition, what nitrous is actually doing is it's dramatically increasing the activity of anterior cingulate neurons. And we've done this with inhibitory interneurons. It doesn't do too much to those, but it dramatically increases in a dose-dependent way the excitatory neurons. And you can obviously plot this and measure it, and these are event rates, and this is baseline under oxygen, and you can see there's a huge increase with oxygen. So it's just the opposite. So clearly we can conclude that a decrease in activity or an increased activity is not a driver of increased pain. Here we increased activity, and clearly it's associated with a decrease of pain. Now, no time to discuss isoflurane, and if I had the time, I could actually get you to what we think is actually going on. We think it's a difference between baseline and evoked activity in response to pain. In the Q and answer period, I said that maybe we can see a difference. But just to give you a little teaser, if you compare nitrous and isoflurane, these are completely different general anesthetics. So as I showed you, nitrous oxide, you have a baseline, and then it really increases dramatically, and it'll eventually return. Isoflurane, you'll get a change that ends up equivalent to baseline by up to about 1%, and then it'll completely shut off, absolutely shut off the ACC. We can talk in what we think is going on. We actually think there's a way to get at a biomarker of pain through that difference, but I don't have time to get into it. I want to switch gears. And the gears I want to switch to is the studies that I've been doing in some DARPA-funded work with Brian Choykett, Brian at UCSF, Brian Roth at University of North Carolina. It's a very heavily funded, large grant to develop polypharmacology, come up with new drugs. And just to give you a hint, it's been one of the most successful collaborations I've ever had. It's a great collaboration. They're a bunch of chemists. I don't know what the hell they're doing. They don't understand anything I'm doing. I don't understand anything they're doing, but it's a match made in heaven. It's really great. They try to teach me chemistry. I did have it before most of you were born, and I don't remember any of it. But it has been successful. So what's the traditional approach? Pharma, what's the old ways of developing drugs? Well, you had hundreds of thousands of compounds, maybe on a computer, but usually on the shelf, and you would test them. And let me just give you an example. We've now identified several interesting new ligands, and of course we're after analgesics, and we're talking now about alpha-2 receptor agonists. Traditional agonists, the ones that we're familiar with that have some analgesic action, but they're sedating, and the classic one is dexmedetomidine. It's a wonderful alpha-2A agonist that is used in the ICU for good reason, because it's not only pain relieving, it's sedating. The anesthesiologists love it. But because it's so sedating, it's not going to be a great analgesic to use as a pain reliever outside of the hospital. The contemporary way is in silico screening. And that's what Brian Choykett and colleagues have been doing for years now. In this particular work, where we identified the royal we, an alpha-2 novel ligand, it began, and this is a paper published with a graduate student, Alyssa Fink, and lots of other people. It was published in Science last year. It began with an in silico screen of 300 million ligands against structure. Now you can do it either against the cryostructure of the target, the receptor, in this case the way it was done, or it can also be done by ligand homology. Nowadays, this is a 300 million screen. Literally now they're doing it with a 2 billion molecule screen. It's quite astonishing. And you have to filter your way all the way down in terms of affinity to structure, etc., PK, whatever. But this one led to this summary where this zinc library of 300 million compounds led to the identification of 4 incredibly potent nanomolar affinities. In this case, and the one I'm going to talk about is what we call 9087. Incredibly effective. And comparing it in analgesic action against the standard dexmedetomidine. So here's dexmedetomidine. Normalized produces an effect in an SNI. This is sciatic nerve injury model and classic neuropathic pain model, where you cut 2 of the 3 branches of the sciatic. The animal becomes extremely hypersensitive in the diaphragmated paw, and you're asking whether the drug can actually normalize behavior. And clearly the dexmedetomidine at 30 micrograms got it up to 100%. But both of the novel agonists will get you twice as twice as good. So it's not only anti-hyperalgesic, it's analgesic. It's getting you above normal threshold. That's a good thing. But more importantly, it's not sedating. Here's dexmedetomidine. Dexmedetomidine at the 30 micrograms is already knocking the animal off of a rotor rod. Go up a little higher to 60 micrograms and the animal basically is not moving. In contrast, the equivalent analgesic dose of the novel compounds are very effective. So there's a lot more of this. We also have a novel NPFF1 agonist, CB1 agonist, 5-HT1A agonist. These are all either published or in press. It's been an incredibly productive effort. Many of these compounds we test with morphine and have seen at least additivity, looking for synergy. The idea to open up that therapeutic window is really what we're after. Maintain the analgesic action, decrease the side effects. Now, how are we testing? Well, everyone knows how boring it is to test. You can put an animal on a hot plate, you can do tail flick, or you can do von Frey testing. And everyone in my lab who does it wishes they weren't in my lab. If you have ever done it. I mean, it's more, I think it's more painful to the investigator than it is to the animal. It's very difficult. And we're always looking not only for evoked pain, we're looking for spontaneous pain. This has been the major criticism in the field. Pharma says, well, you're just studying evoked pain. Fair enough. So now we do condition-place preference, condition-place aversion. We try all these things to get, I want the animal, tell me, how much does it hurt? That's clearly important. Spontaneous pain is what makes people's life miserable, ongoing pain. Well, recently, there was this thing called the black box developed. It came out of Clifford Wolf's lab, and we've been working with the two guys who developed it. They've actually started a company called the Black Box. They recently had a beautiful booth at SFN. Well, it turns out Jared Weinrich in my lab has been doing a lot of software work for them to actually analyze the data. It's been really successful in our hands. It's a simple system. The original paper was published in Pain in 2022. So there's normal transilluminated light in this black box. We now have it where we can actually do calcium imaging at the same time. But then you have Fourier transformed infrared light shining underneath that actually gets us a way to appreciate how much pressure, and it's quantifiable, the animal is actually putting onto that paw. And the first set of studies, we've done many now, I just want to show you a hint of where we're going. This is, maybe in recognition of Ron Melzack, I decided that SNI doesn't happen that often in humans. There aren't too many humans with 2 of the 3 branches of their sciatic cut with scissors. And CFA injections are not that common. I've started to work with the ortho people. They're very interesting. They know everything about bones and knees. My wife just a year ago had a total knee replacement. It worked. But it's interesting. They don't think about nerves. They're studying pain, patients in pain, but their approach is fix the knee. But now we're interested. So we decided it's time to work in something that is so clinically relevant, namely ortho models. We're doing OA models. But in this case, I want to tell you about another model, namely tibial fracture. Now this is just to illustrate a naive mouse, no pain. Can we have the lights down, please? So you'll be able to see, so we're looking now, whoops, backwards. So this is normal mouse, just to see what it looks like. So you can see, here's the infrared light. This is just transilluminated light. And you can see the animals putting pressure on the toes, the pad. They're walking around just fine. It's very cool. Okay? Now, let's see what happens after tibial fracture. And this is unstabilized fracture, which means the fracture has not been stabilized with pins. And remarkably, it still eventually heals, presumably through connective tissue and blood coagulation that brings the bone together. So, if I can get this. So this is now 2 days post-fracture. And I think you can pretty much see that on the right hind limb, the animal's not putting pressure. Is that clear? You're shaking your head. Good. All right? So that gives you an example of what you can see. Now let's look at 11 days. And the animal is now putting pressure on the toes, not the full foot or base of the limb. And this is, again, as I said, it's all quantifiable. That's what Jared has done to find a way to quantify this, because it generates an enormous amount of data. That's what it looks like 4 days as a summary. That's what it looks like at 11 days. That's what it looks like at 18 days. So it's a pretty remarkable, and then by 24 days, this unstabilized, it recovers. So here's the model, and here's a way to monitor behavior. The animal is just moving around and telling you what's going on. And now the question is, what do drugs do? So that's the next thing is we just began some studies, and now we're doing many of them, also with a meniscal tear osteoarthritis model, OA model. This is a control animal where it's had the tibial fracture, and you can see that the animal's not putting much pressure on that back foot. Around here, the drug will be administered. There, now the animal's been administered the drug. And let's move around a little bit. I think you can see that the animal is now starting to put pressure on that paw. This is 5 mg per kilogram, which is, we haven't done a dose response curve, which we have to do. You can quantify this, and we've started to do this, just to give you an example. These are 5 animals, and 4 out of the 5 animals showed clear increase in pressure. One animal, there was no difference, but didn't have much of a deficit to start with, which was interesting. It's just to give you an introduction as to where we're going, because we can do all kinds of drug tests. We're doing knee models, itch models, a whole variety of tests where this is 30 seconds per animal, and they have 1 box, 4 box. I'm not trying to sell them. We have a 4-box one, and 30 seconds is enough time to get data from each animal. It's pretty exciting. Okay, the problem of adverse side effects. This is a question, how does the ibuprofen know where to go? Now, you give this drug, and you get an effect, but any drug, what's wrong with opiates? It's the best drug you can get, but it has adverse side effects. Well, the med students, no, it was actually the graduate students in the quantitative biology program, started something called TikTok for Science. I assure you, I know nothing about TikTok. I knew nothing about TikTok. But they said they asked a bunch of profs a question, and they asked them to answer the question based on a question that I asked med students, and to 30 seconds, 30-second answer, and they'll put it on TikTok. I'm game, sure. So I dictated the 30 seconds. And I showed it to my wife. Now, I was married for many years to a very successful cancer biologist who died 20 years ago of ovarian cancer. I remarried to someone who believes in every alternative therapy ever invented. Not a scientist. And so I showed it to Marcia, and she said, boring. I said, what do you mean boring? It's the answer. She says, yeah, but who cares? It's TikTok. Okay, what do you want me to do? Well, she knows I write poems for all of my lectures. She says, go away and write a poem. Fine, so I went away, came back the next day, wrote a poem. I read it, I said, how's that? It's better, not good enough. I said, what the hell do you want now? She says, you need to wrap it. Okay, I mean, I'm game, I'll go anywhere. So here goes nothing.
I feel no to go through my head.
Well, the answer is the ibuprofen has no idea where to go. You see, you injure your knee, the knee is inflamed, and boy, it hurts like hell. So you take a pain reliever that you hope will make you well. Into your mouth go ibuprofen pills, but they're not looking for your knee. The pills will travel everywhere, of that I guarantee. You see, the ibuprofen has no idea where to go, and so I must advise, ibuprofen will work everywhere, so stomach and other side effects could definitely arise. You see, it's the side effects from oral ibuprofen that limit what dose is needed for your knee, and taking more pills won't solve the problem. This your stomach will certainly agree. An alternative is to treat the injury directly. For example, a medicated plaster placed on the knee, side effects less likely. Good luck, you're now pain-free. But it's a message, and it's why I'm a big believer in developing drugs that stay in the periphery. You want side effects, get them into the brain, and you're going to have problems. And I'm going to do this because I hope I have enough time. How many people here have had diarrhea? Yeah, it's the same as it's interesting. The medical students at UCSF have among the lowest incidence of diarrhea of any group I've ever met. I asked that question, 2 hands go up. We had 3 here. Obviously, everybody has had diarrhea. The question is, what do you take in order to treat? Loperamide. Imodium, right. And you know what that is? It's an opiate. Someone says, it's an opiate? I take an opiate? Yeah, but it's an opiate that's kept out of the brain by transporters. So basically it produces constipation, which is one of the side effects of opiates. It really underscores the value of targeting where the problem is. That's my bias. And that's where a lot of our research is going. Target the area, get around the problem that the drug doesn't know where to go. That was a long intro. Okay, now I'm going to switch to what we're really excited about. As many of you know, I was trained as an electrophysiologist, spinal cord physiologist. My degree is in psychology. It's true, but don't tell anybody. And I did a lot of work at UCL with Pat Wall, spinal cord physiology, mostly in cats. And by and large to date, with maybe 1 or 2 exceptions, all the work is done in anesthetized animals for pretty much obvious reasons. Well, we are now able to solve that problem. And we can record long-term from the spinal cord in an awake animal of the same neurons. And that's what I'm going to tell you how we're doing this and why I think it's important. So this is a fun video. It's a 3-step procedure in order to get the animals prepared. And it's kind of wild, but it's kind of fun to watch. These are all micro CTs. So initially you put the frame on the animal, and there's pins across the laminae. Then we do the laminectomy in a second procedure. And then in the third procedure, we actually put on something that blocks, as you will see, fibrosis, put on a window, and away we go, and we can image. And that's kind of what it looks like. And if you look down, we can actually see we can record now from a large segment bilaterally, the spinal cord, with calcium indicators in, we can record from them. And just to show you, the animals are perfectly fine. They're running around. They're happy with this frame on top of them. So let me give you an example of the problem. Why has it been difficult to record long-term? Well, because when you do a laminectomy, you get fibrosis that happens within days, and then any calcium imaging, you can't see anything. Well, we've solved that problem. And this is 1 month. And it's a Teflon-based system that Andrew Crowther came up with. 50 days, we have now recorded for 1.5 years from the same spinal cord, from the same neurons. And there's no fibrosis. It really has made a huge difference. Just to give you an example, again, I think with the lights down would be so much better. Thank you, that's perfect. So this is an anesthetized animal. So this is like old time, but it could be a month, 2 months after surgery. And I think you can see there's a left pinch, right pinch, and you get lots of neurons. This is GCAMP6, usually driven off of a Cree driver or sometimes by retrograde expression. We don't like injecting things into the cord. So this works just fine, lots of neural activity. This is a fun possibility. This is not single cells. This is integrated activity in the animal, stimulating looking over lumbosacral spinal cord, stimulating different parts of the body, left hind paw, the back, tail base, left or right. You're mapping topography, and someone says, oh, we already know this. He says, yes, we do know this, but one of the things Pat Wall did, and when I was an undergraduate or graduate student, I studied collateral sprouting with the godfathers of that, Chambers and Liu, where after injury, there's a reorganization that occurs in the cord. Well, with this, we can actually see, this is the normal map now of activity from the body. And now what happens when you have peripheral nerve injury, this map completely changes. And because you can monitor over time, you can see the development of the change. It's something that we couldn't do before unless you did it cross-sectionally, and it was extremely difficult. Now, the key thing is, as I tell the students, the brain doesn't care about interneurons. Now, I apologize to people in the audience who study interneurons. They're important, I don't deny it, but the brain hasn't got a clue what the interneurons are doing. All the brain can do is make a decision based on projection neurons. And I was fortunate, so we're interested in the cells that receive input from the periphery, cross over, and via parabrachial or spinothalamic direct pathways, spinal-reticulothalamic, send information to the brain, and the brain makes a decision. Is it an itch stimulus? Should I scratch? Or is it painful? Should I pull away? The interneurons are obviously feeding onto that, but it's the projection neurons. And so I was lucky to have been the external examiner on a thesis of Brian Room in Artur Cagne's lab in Montreal, partly because I speak French, because I grew up in Montreal. And I found out about this FOX2A mouse they'd been working on a little early, and I was able to get it. Now you're going to hear more about FOX2A at this meeting, and it is a dynamite marker. It's a transcription factor that drives expression pretty much exclusively at spinal level in all projection neurons. And if we use a FOX2A Cree reporter and look down on the cord, this is what you see. These are all superficial laminae projection neurons. And high power, this is what it looks like. And these are going to be the cells from which we're recording. We're going to put GCAMP6 into these cells. We can't go deep. That's one of the limitations of the calcium imaging, unfortunately. We're stuck superficially, and we can talk later how we're hopefully get around that problem. Now, so here's an awake animal. Same as I showed you before, stimulus and locomotion, and now there's a stimulus, whoops, come on, nope. Don't be stubborn. No. Doesn't want to play. Well, guess you're going to have to believe me. Anyway, there's a stimulus and there's activity, and you can see activity whether you stimulate on the left or the right. One of the interesting observations, you can see the cells here, is that a stimulus on the left, we're convinced, is also getting activity nowhere near as much, but on the opposite side of the cord. The two sides are connected, even at the superficial laminae 1 level, which is a big surprise to us. Long-term imaging, one of the things in the cord moves like hell. Not only respiration, but the animal's awake, so the animal's running around. Stabilizing has been something that Biafra has succeeded in doing, and you can actually lock on to single axons. Now, why is that important? That happened to have been a Thigh-1 animal reporter, is that if you're interested, and some of the least productive work I ever did was in spinal cord regeneration. You know, after you see an axon go 10 microns after 2 months and you're all excited, we can follow the same axon literally after injury and see what it's doing. Where is it going? Who is it talking to? Because we can follow the same axons over time. This is perhaps the most important thing. The anesthetized animal is different from an awake animal. Here's some examples of left and right spinal cord, cold, pinch, heat. This is just an air stimulus, sound, cold, pinch. And clearly you get responses. In an anesthetized animal, cold stimulus, A lot of neurons respond, pinch, some respond. Even with an air stimulus, and this is interesting, lamina 1 cells are supposed to be nociceptive, but in an awake animal, they're getting input from above that's facilitating, so they can actually respond to innocuous stimuli. But look at the difference in an awake animal. This is the same animal, same stimuli, and clearly the first thing you notice, there's spontaneous activity. There is none in the anesthetized animal. It's 2% isoflurane, maybe 1.5%. And then enormous responses to the same stimuli. So clearly, you're working in a different animal. And then, of course, one of the big questions in the field is the labeled line versus polymodal. Now, Bud Craig, who is a key player in this field, sadly died recently. He went to his grave believing in labeled lines. And fortunately, we never got into a fist fight, but we had a lot of alcohol together discussing the question. I'm a polymodal person, but let's see what happens now that we can do this in an awake animal. Well, here's just one example, not ideal, where many neurons, and there's different stimuli, I don't know if you can read, cold, 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 I can barely read it, pinch, heat, and if you go across, what you can see is there are many neurons, and I drew a line through what appears to be neurons that clearly are polymodal. They're responding to the different stimuli. It's not that clear, and so we took a different approach, different way to look at polymodal. So here's now a series of cold responsive neurons, and what you're looking at, this particular population has a good response, not a great response to cold stimulus to the paw. This is spontaneous activity. There's a little bit, not much. It is an awake animal at this stage. And there's clearly a cold response when you apply cold. So that's fine. It's cold responsive. And that's kind of a summary. The more red it is, the more responsive. These neurons are also heat responsive, even more so. Look at this neuron. If you now compare these, you can see that it's cold responsive, obviously heat responsive, cold and heat. Clearly polymodal, no question about it. Pinch is being added to this, and I don't have time to get into it. An interesting aside, however, there are clearly polymodal neurons that respond to cold and heat. I just showed you that. There's also a population of very responsive neurons to cold. These were curious. Look at these here. Incredibly responsive, big, big response to cold, much more so than the ones on top, which did respond. But these neurons are inhibited by heat. That was a big surprise to us, and we don't believe it's due to heating of the paw. Here's the cold response, and this is the heat response to the same neurons. Here's spontaneous activity. You turn on the heat stimulus and the cell shuts off. Cell shuts off. It's the same cell that was beautifully responsive to cold. It's a very different way of thinking about how a label line can be generated. It's through the interneurons. It's not necessarily direct heat and cold activation, but through circuitry you can generate what are called labeled lines. Remember, why is this important? The brain is making that interpretation, is reading across these output cells and making a decision. What should I do? Was it cold? Was it hot? Was it pinched? Was it a peritogen, etc.? Okay, what's next? Well, this is what's next, and it's pretty exciting. At the moment, the animal's attached. It's awake and running around on this wheel, but it's attached. But now we're actually able to do this with a freely moving animal, with this carrying the miniscope on its back. It's pretty light. Just to give you an example, this is sped up, highly sped up. Here's the animal. Here's the recordings. And Biafra, in this case, is stimulating. You can barely see the stimulus coming across. You can see the stimulus, and there's your activity. This animal is now freely moving, not attached. And you actually get bilateral responses. Bigger on the side of stimulation, you get a little bit on the other side. This is pretty cool. This is now post-SNI. So the animal has had an injury. We've recorded before, but now the animal's had the partial injury, and we're recording now post-injury in this little guy. What you can see, he's happy, he's running around, he's grooming, everything seems fine. But now put him in the field and let him run around And what you notice is we're not stimulating him, he's stimulating himself. There's more activity on the SNI side, so that the cord is hyperexcitable, sensitized, but you still get stuff a little bit on the opposite side. We haven't sort of analyzed this quantitatively, but we could break this down and isolate these single neurons. But it really gives you a sense of where we're going. Lots of questions. What about itch? Everyone asks. We're starting to do this. We're hesitant because when you stick a peritogen in, you screw up the skin, and then we have to wait 2 weeks before we can do anything else. But just to give you an idea that yes, we can pick up responses. We're actually, Bob Lamott sent us some cow hedge, which is great, so that we can actually use these spicules, which don't produce as much inflammation. And this 500 micrograms histogreen, which is a pretty good response, you get a lot of scratching, we can get some activity. Not very much. The question is, do the neurons that respond to this also respond to the pain-provoking stimuli? That's something we're going to know literally in a few days, hopefully before I get home. Lastly, what other things? Well, just if you're interested in vasculature, this is the kind of thing you see when you can record over time. All right, you look down, you see the midline vein, we can see bilaterally, and you come back 2 weeks later and there's vasculature growth. We see this all the time. You come back 3 weeks and it's gone. We see this all the time. There's growth and loss, growth and loss. This is a very dynamic cord that you're looking at. And lastly, we'll talk about microglia, which we're interested in, many people interested in the relationship between so-called nerve injury activated microglia, particularly in males. Well, the traditional way, of course, is to use IBA staining. We do it all the time. And this is an animal that had an ipsilateral sciatic nerve injury. You get a big upregulation of microglia, either more microglia or a different morphology. You see stuff in the ventral horn. That's ATF3 as well, because you cut the peripheral nerve. That's standard. Everyone does that. But by taking a CR3 GFP mouse in the same system now and monitor that, this is what we can look down at microglia. We can actually monitor and see movement in the meninges. It's quite spectacular. See monocytes moving around in the meninges. And this is now recording microglia over time. Same animal. Pre-injury, you don't see very much. Then we do an SNI at this time, and you wait. And then by day 3, now you start to see ipsilateral, clear, very increase in microglia integrated. And then whoops, it looks like all of a sudden you're starting to see things on the opposite side. This is something you would never pick up in a cross-sectional IBA study because you wouldn't know what your baseline is. You'd never see the evolution. So it's giving us a sense of literally the evolution of injury-induced changes because we can record over time in the awake behaving animal. So that's where we are, just to show the difference over time. Ongoing questions, we're still interested in the label line question. Algogen versus peritogen, how do they interact? Is it the same cells? Projection and response during and after injury, one of the most interesting questions. If you're interested in the transition from acute to chronic pain, wouldn't it be nice to be able to do it in the same animal, not in a cross-sectional way? Effects of analgesics, we're doing a lot of that, especially some of the novel analgesics. And this I write as pie in the sky. It's not pie in the sky, particularly because since we're already recording in the brain, Biafra has done a lot of work in the brain. And so to now, in the awake, freely moving animal, we'll pop a miniscope in the amygdala or anterior cingulate, we can now relate the activity of patterns of activity in the cord to what happens in the amygdala or cortex while the animal performs a behavior. And we're pretty excited about that. This is the group. This is Jared. Cindy's a graduate student. Jared is a postdoc who did all of the initial cortical imaging work. Joao and our colleagues, our chemistry colleagues, have done the in silico screening work. Jared and this group here in the ortho have done the black box work in the fractured tibia. And then all of the cortical and spinal cord imaging is from Andrew, Biafra, or both postdocs, and Mariella, who's a graduate student. And the mice came from Artur and Brian. And of course, we have funding, which we would get nowhere without it. If I have 1 minute to finish up. So I've been doing this for a long time. I was a postdoc in Pat Wall's lab, as you can see. By the way, Rohini, when you introduce me, you should never mention dates. It's too late. But anyway, in 1984, I was a postdoc in Pat's lab, and I was invited to give a talk in Seattle at the IASP meeting, and don't ask me why. That was one of the first poems I ever wrote. And so just for historical purposes, and it's kind of relevant to the topic, some of you may have seen it. It's called Praise the Lord for the Spinal Cord. There are myriad molecular mechanisms that generate pain. Let me name just a few. There's peripheral sensitization, arachidonic acid, COX enzymes, and NGF2. Yes, the nociceptor transmitter is glutamate, but it co-expresses CGRP, providing hopefully continuous NIH funding for a neuroscientist like me. Now, Marx had it wrong. It's the endorphins that are in fact the true opiates of the masses, so let's develop a better opioid, whether it's in the mu, delta, or kappa opioid receptor classes. And what a pity that the discovery process is at times as slow as molasses. Amazingly, we still do not know how good an analgesic drug grass is. What is clear is that pure pathways do not access the brain, which is undoubtedly why anterolateral cordotomy for pain's on the wane. But since analgesic drug action and circuit analyses exploit neurochemistry, brain stimulation, and certainly even transplants in the treatment of pain. For there are few in this audience who could readily afford to relinquish the functions performed by the cord. Sure, the brain is important, but the spinal cord's central. So come, blow your horns, be they dorsal or ventral, and let us end by rephrasing the words of the Lord, honor thy mother, and don't cut the cord. Thank you.
Thank you, Allan, not only for the world-class science, but the sense of poetry and scientific spirit, which was very, very beautiful. So, questions? Yes.
So, the microglia effect you're seeing on the contralateral side, I was just wondering, what's the timing of that in terms of what's comparative happening on the contralateral?
Using this imaging approach, we see it after maybe a week. But with recording, there's two aspects to recording neurons. We will pick up cells and these are all projection neurons on the opposite side in superficial laminae, same day. Many fewer cells. So there is crosstalk. Now, Andrew has been doing some transneuronal tracing studies from FOX2A neurons on, say, one side of the cord, and he is seeing transneuronally labeled, presumably one synapse, on the opposite side. So my PhD thesis was all about two sides of the cord talking to each other and ways for the brain to get information through small circuits. And that's what Andrew is sort of building on with modern technology. I believe that the whole view that pain is a cross system and it's all long pathways is really a misconception.
And that's true for the microglia as well as the neurons?
I believe so, yes. Yep.
Thanks for a great talk. I'm wondering if you could develop your ideas of how you think the processing in the spinal cord works. I mean, you have like this very active heat neurons also that inhibits cold neurons, and what are the very active versus not very active neurons doing, and how do you think this works?
I'll be honest and say I don't know the answer. This is really, in a sense, we're fishing to try to understand what is going on. And of course, we're focused on the lamina 1 cells because those are the ones from which we can record. We're actually starting to use what are so-called nets, these nanoelectron, came out of Chongzi's and Lan Wan's group in Rice, where we can go deeper. These are like hairs, so you can record long term. Clearly, it's a pattern of activity across these neurons that determines the output. So looking at one cell and saying it's cold responsive and maybe even cold and heat doesn't really tell me enough about its impact on behavior. Because the impact on behavior, unless you're talking reflexes, and I think that's more deep cells, is going to be what happens up in the brain. Now, and then context. We are really interested in maybe having a virtual reality system where the animal can start to choose one side or the other to avoid a stimulus or get a drug. But I can't tell you what the significance of any particular type of cell is, other than that it exists in the awake animal, and you'll never see it in an anesthetized animal. That cold responsive cell is purely cold responsive, and you wouldn't know anything about its heat sensitivity. So that's the basic message, is that you do get that. Now the group, certainly out of even in Edinburgh or in Glasgow, but coming out of Sarah Ross's group, where they're doing ex vivo preps, that's closer, but of course then there's no brain attached. And the brain is clearly influencing what's happening in the cord. So this is allowing for the brain to influence at the same time as avoiding anesthetics. So it's going to take time to eventually put this together and make sense of it. It's an important, it is the important question.
There's a question there, right? Oh, then let me ask a question, Allan. To continue along this line that Patrick just mentioned, where do you think that the code is actually decoded, in terms of deciphering what kind of stimulus modality it is? Because in the spinal cord, as you pointed out, we have both specific and polymetal cells, and there's a population code. So in your view, since you're the master of anatomy, where do you think this decoding is happening?
Well, the brain has to be doing the decoding.
And in the brain, the cortex, or because the thalamus sort of mirrors the spinal cord, you think?
Who votes for the amygdala? Who votes for VPL? I happen to be a big believer that pain-wise medial thalamus is really important. Classically, people forget. I wrote this thing recently called, a little review called The History of Pain Pathways, including many long forgotten. And the reason why I talked about that is lamina 7 is the origin of bilaterally responsive spinal-reticulothalamic neurons that have classically, historically been involved in the real pain experience. And they're completely ignored now. They're very difficult to record from because they respond to everything. They're very anesthetic sensitive. Well, I happen to think that those are particularly key. It's the reticular formation that I believe is probably where a lot of the magnitude of the impact of pain is being generated. But the actual interpretation is what is the type of pain. I would say it's got to be somewhere in part up in somatosensory cortex. That's where the quality comes in, the modality comes in, but then the affect that's added to it comes in from, say, insula, parabrachial, medial thumb, whatever. I mean, there is the condition of pain asymbolia, right? Patients with lesions of temporal lobes, they can tell you that's very hot. Yeah, oh boy, that's really intense. Whoa, yeah, but it doesn't hurt. They've completely dissociated the sensory discriminative from the affective. So there are different parts of the brain that are clearly handling these. And if we will not get to it unless we can record from both at the same time.
And so for this, we'll also need some tests that enable the mouse to discriminate and report the discrimination to us.
Yes, I've actually, here's a kind of fun thing, and I'm hoping that Ugo Bazile claims they're going to build this for me. I want to know, I have this great video, which they don't let me do anymore. I show a video, I'm 30 years younger. Put a blood pressure cuff on the arm, blow it up above systolic, blocks all large myelinated fibers and small myelinated fibers. The arm is paralyzed. All the unmyelinated fibers are functioning totally normally. They're cold fibers, whatever. But if you pinch, what do you get? It burns. Ice, it burns. Heat, it burns. Pinprick, it burns. Neuropathic pain, ongoing burning pain. Okay, now SNI, cold stimulus to the mouse. It avoids it. What did it experience? Did it experience burning or cold? And so they're going to build this apparatus for me where the animal is trained. If it wants to turn off a hot stimulus, it's got to go to this nose poke. If it's cold, to this nose poke. If it's mechanical, this nose poke. And then see which nose poke it goes to. When there's a cold stimulus in a setting of nerve injury, does it go to the hot button to say, that's hot? I don't know, but we don't know what the animal is experiencing. We can say the animal's avoiding something, but I want to actually know to what extent they're experiencing what the humans experience, and then try to relate that back to the firing properties in these animals. There was a question up there, I think, yeah.
Can I go first?
Yeah. Yes.
Mike.
Yes. Yeah.
It sounds really fun in your lab, I have to say. Can you hear me?
Yep.
So I'm interested by this idea. You really are focused on the peripheral as a treatment space. Clearly, affect is not a peripheral problem. It's not a peripheral part of chronic pain.
What? Affect.
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 Do you think that you will treat affect from the periphery?
Indirectly. Obviously not directly. Take a patient with postherpetic neuralgia, and I saw a lot of these when I was a postdoc with Pat Wall. I used to go to the National Hospital and saw patients one day a week with Peter Nathan. I wanted to see patients, and I send my people in my lab to the pain clinic with the pain physician. I want them to know that a mouse is not a human, number one. So you have a postherpetic, first division postherpetic, burning, pain, horrible, hypersensitivity, they're terrible, their life is ruined, local anesthetic injection, and 100% of patients, pain goes away. In some patients, it'll go away for 3 weeks. In most patients, it only goes away for the duration of the anesthetic, say bupivacaine, 8 hours. So there's a central sensitization component that accounts for the prolonged effect, but it always comes back. So the affect is gone by eliminating the input, but I'm not affecting the circuitry of affect. Okay, so I think one drives the other. Now, in cases of axial low back pain or even fibromyalgia, is that a purely centrally based problem that targeting periphery is not going to help? I have this big discussions with Jane Ballantyne, and we're still friends, surprisingly. But it's really a question of when are opiates appropriate. So affect is a product of the brain. It is not a product of the periphery. The question is, what's the driver of the chronic pain? And the two are usually linked, but not always.
So it would be then about triaging. Well, you could do that easy test, do a lignocaine sort of thing. If it works, then go for a peripheral drug.
You pointed at your head. Do you mean lignocaine in the brain?
No, well, because you were saying, in your example, you were saying injecting it around the eye, right?
Right.
Yeah, so you're saying, yes, it will work peripherally, except when it doesn't.
Well, yeah.
Yeah, that's fair.
Well, take a phantom limb pain patient.
Yeah.
You inject the stump. The stump pain goes away, the phantom disappears, and the phantom limb pain goes away. But we know from Herta-Floor's work that there's a reorganization of the brain in the setting of phantom limb. Everybody has a phantom limb. Some people have pain, but there's a driver. And the question is, under what conditions is there no driver? That's what I don't know. Are there conditions where what I'm hypothesizing doesn't work? Although I do believe that the major problem is the drugs don't know where to go, and when you give systemic drugs, you're increasing the likelihood of a therapeutic window problem. You're going to reduce it by targeting the periphery to the extent that you can.
So Allan's going to be here for the entire meeting, which is wonderful. So there's lots of time for discussion. We'll take one quick last question, please.
Okay, I think it's going to be very quick. It's related. Hi, Allan. Over here. Thanks. In relation to this affective question, if I understand correctly, your hypothesis is that the nitric oxide affects only the affective component. And so have you ever tried looking at your spinal cord projection neurons under nitric oxide and see whether or not you get subpopulations that are affected?
To some extent, we've just started. We've done, for example, FOSS studies in the cord with, for example, this is Cindy Liu's work. Nitrous oxide in a formalin test model doesn't block licks. I mean, it blocks licks, but not reflexes. So in that model, it does similar things. If we look at FOSS, it doesn't appear that there's an inhibition of the lamina 1 cells. So it appears that the effect on affect is probably supraspinal. It's a really important question and one we're trying to get at. What we believe is, and it's based on the isoflurane data, is that here's your baseline activity. Nitrous increases baseline. So now baseline's here, and you throw in the laser and it goes up a little bit. So that's the difference. With other anesthetics, the baseline doesn't change. At 1% isoflurane, the baseline is exactly the same. You get to the same level with the laser, there's more pain. So we believe it's the difference between baseline and evoked that determines the magnitude of the response, not just the total amount of activity. It actually gets into something called connected consciousness, which is the real fear that in some patients under general anesthesia, they actually experience 1/20th of patients. You can experience, they won't remember anything under anesthesia. You can do it by putting a blood pressure on the arm and having the arm talk to you. And half of those 1/20th, so 1/40th, experience pain during surgery. That's pretty scary. They don't remember it, but it really has to do with how much do you have to shut off the brain in an immobile, apparently unconscious person to actually block the pain experience. We think there's a biomarker up there that might give us a clue as to how better to get rid of that. That was said a lot. That would have been a 20-minute lecture. I'm sorry. But thank you very much.
Thank you, Allan, for this very stimulating.